So, what's the latest news? Well, we have now infused the first patient with a autologous cell therapy, which is intended to cure HIV. So we are in the phase 1 right now of a clinical trial for a cell therapy that we've been developing now for many years. And we expect to see safety data on that sometime this summer, and efficacy data on that, an efficacy signal by the end of the year. So this is an exceedingly exciting time for us right now. As you know, at previous bios, you've seen me come out and announce that the IND was submitted, the IND was approved, and now we're at the point where we have already recruited 6 patients. We have leukapheresed their blood samples. We have modified those blood samples to be HIV-fighting T cells and that are immune to HIV, and we've actually reinfused the first patient with that product. So as of today, that patient has received a couple of follow-up checkups, and they have not observed any outward adverse events. So as you can imagine, the company is extremely excited right now. I'm so happy to be able to report this to you. And actually, by the time you're watching this, there's probably even more news out there. So I hope you'll connect with us on social media or look at our website, at our press releases and articles and so forth. I think things are going to start coming relatively quickly on this. And, keep your fingers crossed. I am highly confident that we will be able to make a functional cure for HIV. And one of the reasons I'm so confident right now is because of the importance of this safety data, right? If we don't see adverse events in this first patient, if we go on and treat the second patient shortly thereafter, and we get all these 6 patients by the end of the year, if there's not an adverse event, at least we know the process is safe. Now, I believe that we are returning a cell product to the patient that's potent enough to suppress their virus for a lifetime. In other words, they will never need to take antiretroviral therapy again. They can never get AIDS. They can never infect another person. And also, because these cells will persist in their body, we believe, for life, they can never be reinfected with HIV. Obviously, this is the dream come true of HIV patients. And when you look at the cost of the process that we put together, it looks like we could make it cheaper to cure HIV patients than to treat them for life in insured markets at first. And given the trends in gene and cell therapy, it looks like the capabilities of gene and cell therapy are on the rise. The costs are coming down. So there's good reason to believe that over time we can bring the price down, raise the efficacy of this, and who knows how long it'll take, but I'm hopeful that we may see an end to HIV in my lifetime, in our lifetimes. So anyway, that is all speculation to a certain extent, but I gotta say, I am exceedingly confident about the potency of this product, and let me explain why. So first of all, we have the dream team of HIV researchers and doctors associated with this project. That's been true from the start. And so a lot of my confidence up to now, because it's all been preclinical data and things like that, has been that I understand the basic process and it makes sense to me, but it's these top-notch doctors, these top-notch researchers who have remained excited about this entire project throughout the development of it that has really continued to buoy my confidence in our ability to get to this end goal. So, of course, that's been, fundamental to the momentum of this project, to have those fantastic people on it. But it's also been something that helps the CEO and even people in the community to keep hopeful about where we can get to. So, but then recently, you may have heard this already, but what we were able to show in the preclinical data is that when we make this autologous cell product, so we're taking T cells from an HIV-infected individual's bloodstream, We are modifying them to be resistant or immune to HIV. We're amplifying them up to 1 billion cells and putting them back into the body. And before the clinical trial, of course, we couldn't put that back into the body, but we could make that blood and test it ex vivo. So we did that, and when we challenged it with HIV and latently infected cells or HIV-infected T cells, what we saw was remarkable clearing power of that blood. A remarkable ability and an unusual ability for that blood to clear itself of HIV. It was so remarkable that we showed it to NIAID. Of course, you know, we're in Rockville, Maryland. NIAID's right down the street. Fortunately, we know a lot of the folks there. And Tae-Wook Chen took an interest in this. Tae-Wook Chen is the head of the HIV lab at NIAID. And he wanted to reproduce that result in his own lab. So we gave him the reagents. We gave him our lentiviral vector. It was completely arm's length. We let them go ahead and repeat our experiment on their own patients, on their own blood draws from HIV-infected individuals, and they got the same result. So that was tremendously exciting that they agreed to go ahead and validate our experiment. And what that turned into was an article in Molecular Therapy that anybody can go ahead and read. You can just look it up online. But it looks to folks that are reading this article that perhaps this treatment is 3 to 5 times more potent than it needs to be to create a functional cure. That's not a guarantee, but the numbers are exciting. All right. So what else is making me confident about this product? Well, part of it is what I mentioned already, 1 billion HIV-specific T cells. So we're collecting about 1 million T cells from the patient's body, and we're isolating the HIV-specific ones. And then we modify them to be immune to HIV. What that means is that an HIV viron does not have the normal advantage over the T cell any longer. So an HIV viron attaching to this T cell or attempting to attach is unable to infect it. It's unable to cause that T cell to become part of the body's infection and start spreading the infection around. And instead, the T cell is able to kill that viron the way it's supposed to. All right? So we have changed the dynamics of HIV with respect to these T cells, in that the T cells now have an advantage over the viron instead of the viron having an advantage over the T cell. And that means that the virons, the HIV virus, cannot attack and deplete a person's natural immunity to a viral pathogen, in this case, HIV itself, right? Because if HIV cannot kill the T cells that are out to get HIV, the T cells act normally and they are able to clear HIV virons like your cold T cells are able to clear a cold. We've removed the advantage. Okay, so once we make that modification, the next step is to culture it on up. So we go ahead and we get those T cells to reproduce themselves until we have about 1 billion of them. And this is a very interesting number because a lot of the virologists and immunologists that you know, or maybe you are one, will generally say that after a viral pathogen is cleared from your body, the number of virus-specific CD4 positive helper T cells that are in your body are about 100 million. So we're actually culturing up 10 times that because we think that even with the cell infusion, enough of them will survive that you have more than you need to create a memory level type of protection against HIV. That's what we're trying to do. We're trying to restore the body's natural immunity to HIV. It seems to be working out vis-a-vis the numbers, but also the theoretical data and the preclinical data that you've seen, you saw in the NIAID study, if you go ahead and read that online. All right. What's another reason why I'm confident? Well, we are doing we're on the arc, let's say, of this whole HIV cure story. It started with them discovering that CCR5 was the entry point on T cells, and then there was the Berlin patient experiment where an HIV-infected individual, Timothy Ray Brown, showed up with an incurable leukemia. And so what they did was they gave him a bone marrow transplant, and the doctors thought, let's do an experiment and let's transplant from a donor who's CCR5 negative. And what happened is that Timothy Ray Brown became functionally cured. So he didn't need antiretroviral therapy to suppress his own virus. So that's the goal. But of course, bone marrow transplants aren't going to do it. They're expensive and they're dangerous. About 6 out of 10 people that receive a bone marrow transplant will die from the trauma of the transplant. So that's not a long-term solution. So that experiment got repeated. There's been a number of people that have been cured with bone marrow transplants. But then I think a big step forward was when a company called Sangamo decided that they would try to create that CCR5 negative state of T cells in a normal person with HIV without doing a bone marrow transplant. So you understand that with gene and cell therapy, you can go ahead and modify cells. That's what we're doing in our program, so that you can knock out the expression of certain genes. And they use something called zinc-fingered nuclease. That's a technology that Sangamo pioneered in order to clip the CCR5 genes in bulk T cells that were removed from the patient. So what happened to them? Well, turns out that in the case where a patient already has one of their alleles negative for CCR5, Sangamo was able to get the other allele or enough of them so that somewhere around 1 in 10 of the patients in the study were in durable remission. As a matter of fact, we know of one that has been in remission for 7 years now without antiretroviral therapy. So that's a tremendous outcome. What it says is that gene and cell therapy might be a substitute for bone marrow transplants. But when you compare Sangamo to our current experiment, what you find is that their best patient, so if you look at the number of CD4 positive helper T cells, the conductor of the immune orchestra, the cell that is the specific target of the HIV viron that is depleted in the first 90 days of acute infection. Well, their number compared to us, or let's say our number compared to theirs, their best number compared to our worst number in our study, ours was 2,000 times their dose. All right. And what's important is that we were able to get that dose on less than 1/10 the vector cost. So they were getting 1 in 10 patients. We're 2,000 times the dose. And so I'm feeling optimistic that we can cross the threshold of functional cure for a large percentage, possibly even all of the patients. Again, time will tell us. And I think we're going to get some signal, at least from these initial patients, that will give us some indication that these cells are in the body and working as expected. All right, so I already alluded to this. The schedule is that we believe that in July, the DSMB will meet and they will read the reports, the data from the first patient, and we're hopeful that they'll give us the thumbs up that it looks safe. And then we will continue with this study throughout the year. We should be able to treat the first 6 patients by December. And since we're collecting blood samples all along the way, we should be able to demonstrate that our T cells are in there, that they're fighting HIV, that they're surviving, that they're healthy, and that they're staying at those levels. And that is the kind of data that would give us an efficacy signal. We could even see an aberration in the viremia level that could be attributed to those cells. So if normal viremia, even at undetectable levels by standard lab tests, were to lower even further, and these are things that can be measured in special tests, well, that would be exciting data. But of course, the thing that we want to power through to is once we have safety, we want to do another phase 1 or a phase 2 that will require an analytic treatment interruption so we can see, are these patients virally suppressed and free from the risk of AIDS and at levels that are below what they need to be at to be completely uninfectious? Well, we can see that by requiring patients to do a treatment interruption on their antiretroviral therapy. So that's in the future. That's the gold standard of seeing whether this works. But I think we're going to get what we call efficacy signal in the meantime. That's obviously very, very important, and it would be very exciting to the community and, of course, to us to start seeing some indications even in the blood that this seems to be working. All right. Now, we're not just an HIV company, because what we are is a gene and cell therapy company that was founded to create the most efficient system possible for designing and developing new drug candidates. All right. So we are using our own system to make this HIV product. But what we're seeing is a tremendous efficiency in getting from idea to first human efficacy, or into the clinic and start getting the safety data, that we believe is ahead of other companies, and substantially ahead of other companies. And the way that we do it is that we have a platform of reusable, recombinable components that you can mix and match that can form a foundation for a myriad of diseases. So what does that sound like? It kind of sounds like an iPhone that does all of the critical work of, in our case, delivering the genetic constructs, expressing them at therapeutic levels in a safe manner, with targeting that keeps the expression of these genetic constructs in the cells that need them and protect healthy cells, or cells that don't need the treatment, from that drug. That's the beauty of gene and cell therapy. That's what we're doing. These components do a lot of that heavy lifting. And we think that one day, curing a monogenic disease may be like writing an app on an iPhone. Once you know exactly what needs to be done, once you're intimately familiar with the disease, you should be able to tell us, here's what we need to knock down, here's what we need to overexpress, and here's where we need it done. And we would be able to, with a thought experiment, tell you whether that's possible with our existing platform. And we're going to keep pushing that platform forward. And in fact, it's already quite expansive, and it includes monogenic diseases and even projects in immuno-oncology that can address solid tumors. All right, so this has been a long presentation so far, but I do want to really quickly just sort of bring you through what we're doing in immuno-oncology. That could be the second act for American Gene Technologies. If you start to see efficacy indications this year, or if you actually see first human efficacy, that doesn't just prove that HIV is curable, it proves that the platform works. It proves that we've created a more efficient system where we can start to turn out a much accelerated pipeline of a variety of chronic disease solutions, monogenic disease solutions, and even immuno-oncology cancer solutions. All right, so let me just tell you real quickly how our current gamma delta T cell activation, stimulation technology works on epithelial solid tumors. So epithelial solid tumors, you may know, are things like breast, prostate, lung, liver, colon, kidney, ovarian, pancreatic, and head and neck skin cancers as well. You probably know these are some of the most deadly solid tumors around. And in fact, 900 people a year, I'm sorry, did I say a year? 900 people per day in the United States die from that range of epithelial solid tumors that I just mentioned. Okay, now normally you have a surveillance system in your body called gamma delta T cells. Type 2 gamma delta T cells are the key ones. And they have a gamma delta chain antigen receptor that attracts them to areas of rapid division in your body. And then they have an NK receptor on them that will go ahead and it can sense stress molecules on the surface of cells that indicate malignancy. And when that NK receptor triggers, the cell becomes cytotoxic and kills, that T cell will kill the cell that is displaying those stress molecules. Okay, so this is how you actually stay clear of epithelial malignancy and don't grow a epithelial solid tumor until you do. And 1/3 of people, unfortunately, will grow an epithelial solid tumor in their lifetime that needs treatment. And fortunately, because these cells are so good, 2/3 of us will not encounter that problem. So that's not bad odds, but for the 1/3 of people, that's a lot of people on Earth. What are they going to need? Surgery, radiation, and chemotherapy. But do they really? Well, we think we might be able to change that equation. So how do we do it? With some bad luck or just, sort of normal reduction in your immune system over your lifetime, normal exposure to different types of damaging chemicals, radiation, stress, diet, everything, right? You may get enough malignancy in one tight area where it might get ahead of those gamma delta T cells. They're good at eliminating a few cells here and there and preventing a solid tumor from erupting. But once a solid tumor is erupting, you can imagine it's blowing up like a balloon, and they just don't have the power to eat that away at the rate that it's growing. Okay, so here's what we do. We go ahead and we put a small amount of a lentiviral vector that we call immunotox into the primary tumor. And what that does is it modifies a small percentage, maybe we think about 20% will be enough, of the tumor cells themselves. So the lentivirus is going and infecting the tumor cells. It implants a genetic construct that causes the overexpression of IPP. This is a stimulatory small molecule that binds to the microenvironment, and it creates a site of immunogenicity that attracts all those gamma delta T cells in and activates them. Okay, so the gamma delta T cells come in, they end up coming into this environment. Not only do they get activated, but they proliferate as well. So what happens is your body starts ramping up against the cancer, almost like the way your body ramps up in the case of a vaccine or a virus, right? You've got T cells now that are becoming highly activated, looking all over the body for that malignancy. Well, of course, they have the power to go ahead and attack the primary tumor that we've just infected, and that tumor is obliterated. But what we're also finding is that because they're circulating all over the body, they're finding untreated tumors. So your secondary tumors and your metastases are also being destroyed. And we have a 1,000-mouse study where the mice are coming back, 85% of them completely cancer-free from advanced stage human cancer, and using human gamma delta T cells in the mouse. So I think that this mouse model that we have may translate well to humans. Humans have plenty of gamma delta T cells, and if they don't, gamma delta T cells are allogeneic. So we can actually take gamma delta T cells from somebody else and put them into a patient that needs them, and we can make them look a lot like the mouse. So it may be the case where an intratumoral injection will light up that primary tumor sufficiently where the natural gamma delta T cell response will clear not just that tumor, but it'll do a sweeping of the entire body for epithelial malignancy. This would mean that it wouldn't just catch the metastases of that particular primary tumor. It really would act on all epithelial malignancy in the body. So it would have kind of a flushing effect. All right, this is further off than our HIV project. It's not in the clinic yet, but we have a great collaborator, Dr. Dean Felsher from the Stanford Medical School, and we hope to get into the clinic in 2023, into his clinic and other clinics with this experiment. And we think that maybe the first target will be liver cancer because it's fairly easy. Even though it's hard to do an intratumoral injection, what we're seeing is that we can do a cannulation of the liver, which is normal in liver cancer treatment. And we can squirt out vector near the primary tumor, and it looks like there's enough uptake in that primary tumor that it should start this host-wide, body-wide reaction. This is really exciting. The dream of the company when I founded it back in 2008 was to send radiation and chemotherapy the way of bloodletting and leeches. And I got to say, this is exciting stuff. I'm really hopeful that we may see that. All right. Well, I want to thank you for listening to the presentation today. I want to remind you that our name is American Gene Technologies. We're at americangene.com. We have all sorts of social media. We'd love to have you hook up with us and follow our story. And thank you very much for tuning in.